moving towards the genes, we have the introduction for the genetics. We have introduction the DNA, RNA, structure of the RNA and the DNA function, and the human genetics. So first we are moving towards the nucleic acids. Nucleic acid molecule that store information for the growth and the development. So there are two types of nucleic acid. We have the DNA and we have the RNA. DNA is basically deoxyribonucleic acid. They both are basically composed, basically contain sugar in the structure. Why we call the deoxyribose? Because the DNA misses the basically OH, or you can say hydroxyl group at the position number 2 in the structure. So we have nucleosides. Nucleosides are the structural subunits of the nucleic acid, the macromolecules that convey genetic information into the living cells. They contain of the nitrogen containous bases. These are basically the nitrogenous bases, which are basically purine and the pyrimidine, and and these basically contain further our adenine, guanine, cytosine, thymine, and in the case of the RNA, you can say uracil. So nucleoside, what are the nucleoside? Nucleoside basically are the sugar and the base, and when nucleoside basically contain the phosphate group, it is called a nucleotide. So our DNA is basically the nucleotide structure. Nucleotide are the building blocks of the nucleic acid. They are composed of 3 subunits. A nitrogenous base, you can say that the nitrogenous base is here, a 5-carbon sugarine, you can see that, and they are also known as the phosphate nucleotide because it's containing the phosphate in their group. What are nitrogenous bases? These are the basically purines, adenine, and the guanine, and pyrimidine is the cytosine, thymine, and in the case of the RNA, it is the basically uracil. You can see the structure of the purine and the pyrimidines. The structure of 2 most common purines are the adenine and the guanine. The structure of the 3 most common pyrimidines are the cytosine, uracil, and the thymine. So we have pentose sugar. There are 2 rigid pentose sugars. RNA contains the ribose and DNA contains the deoxyribose. As I have already told you that they both basically contain the sugar in their structure. The sugars have their carbon atoms numbered with the primes. Primes mean the lowest one comes first to distinguish them from the nitrogenous base. So you can see that. Ribose into the RNA 1, 2, 3, 4. We have the structural arrangements, and ribose into the DNA. You can see we have the hydroxyl group here, and we are missing hydroxyl, only hydrogen, not the hydroxyl group. Oxygen is missing, that's why it is only the hydrogen group. Nucleotides are the basically phosphate phosphate containing groups, and we have listened about what is the ATP, ADP, and the AMP. ATP is basically the energy production phosphorylation. It means when your body wants to work, wants to do work, or wants to do exercise, or you can say exert its own energy. So it needs something from your body. You can say your mitochondria is the basically powerhouse of the of your cell that basically produces the ATP. So why we need ATP? We need ATP for the energy production mechanism into our body. For example, if you are taking food, and food containing basically carbs, and you can say glucose. So we have to break down the glucose into our body for further functioning and distribution. It's a glucose in the different parts of the body for the further functioning, and some area it can be stored when the body is basically consumed it. So in the glycolysis, we can see that at the different steps, ATP is basically converting into the ADP. So what is the ATP? ATP is an adenosine triphosphate. When there is an energy metabolism mechanism, this ATP releases its the phosphate and converts into the ADP1. Okay, and when and again the process, ADP is converted into the AMP. So it's mean from the triphosphate, it is converting into the diphosphate, and from the diphosphate, it is converting into the monophosphate. So you can say we have the AMP, adenosine monophosphate, adenine, and the ribose, and the phosphate. It's basically containing the adenosine monophosphate, and additionally phosphate link. If we had 2 phosphate, then it is the adenine, ribose, and the phosphate, 2 phosphate, and we have the 3 phosphate. It is basically adenosine triphosphate. So we all need for the energy production in the human body. Function of nucleotides. We have the nucleic acid biosynthesis, energy production, and the transduction. Protein biosynthesis, regulatory cascades. What are the regulatory cascades? And the one of the regulatory cascades is your apoptosis, where your body cell have to die. We all know that we have for example, we have the RBCs, and every RBCs have their own lifespan. That is basically 120 days. So where they go when they are, when the 120 days have passed? So what is their process? Or we can have question in our mind that what will happen with the RBCs once they are into the human body? So we need some mechanism into our body. For example, we have a food in our fridge, and we know that for example, we have milk in our fridge, and we know that after maximum after 2 days it can be contaminated, or it can have some different of the bacteria, and you can say sometimes you can feel the smell into the food. So once your food is basically destroyed, so you have to discard your food. So that's why when your RBCs are basically, and once your RBCs are in your body, so where they will go? So after performing their function into the body, they are basically consumed, or basically destroyed through the process of the apoptosis in your body. And after their breakdown, their cell is basically break, and their mitochondria is basically break, and the dead bodies of the RBCs are basically stored into the human body. Some of the part is basically stored into the liver, some into the spleen, and as because spleen and the and liver basically store that bodies as their for their nutrition, and excess of their dead bodies is basically excreted through the urine and through your you can say excretion pathway into your body. So this is the basically regulatory cascade. So we have the intra and the intracellular signal transduction. That is the basically our receptor mechanism in our body. So how our receptor work into the body? We have intra and intracellular signal transduction. We have the 4 type of the receptor signal transduction into the human body. We have the biosynthesis of the some biomolecules. So we can say that when food is break down, it is converted into the different other products. So this is the biosynthesis and some biosynthesis of the some biomolecules. So I'm not going to explain all this. I'm just giving you some example relating to these. What are these? So we have the DNA. We all know that DNA is basically discovered by the Frederick Griffith. That is a factor. Okay, first we will focus about that discovered into the bacteria by the scientist that was the Frederick Griffith in the 1928. Then Rosalind Franklin, that was the first person. She was the first lady who basically find out the X-ray crystallography structure of the DNA. So in the history, Rosalind Franklin is basically called the DNA inventor, or you can say she was the first who basically discovered the structure of the crystallography structure of the DNA. But later on, Watson and Crick basically, they claimed that they discovered the X-ray structure of the DNA, and we have a contradiction to the history about all these things. So we are not going to discuss here, but in the history, Rosalind Franklin was the first who basically discovered the crystallography or the structure of the DNA. That so we have Watson and Crick proposed. So what did they propose? They proposed that DNA had specific pairing between the nitrogenous base. That's the basically adenine and thymine. We can see A and T, and cytosine and guanine. It is another pairing, and DNA was made of the 2 long strand of the nucleotide, and in the specific way called the complementary rule. Complementary rule, it's mean that adenine have to bind with the thymine, and cytosine have to bind with the guanine. But in the case of the RNA, thymine will be replaced with the uracil. So it's the basically complementary mean that it has to bound like an adenine thymine. If we talk about a DNA mutation or genetic mutation, it happens when adenine by mistake binds with the guanine, cytosine, or maybe additional adenine. That is the basically DNA mutation or genetic mutation that can vary the structure of the human human genes. So you can say single replacement or single defect into the any gene can be harmful for the human body, or sometimes it may be beneficial. It depends on the physical and the structural appearance and some kind of the disease disorder. So we have the structure. We have the rings of the lighter nitrogenous bases. You can see these are basically legs of the lighter phosphate and the sugar backbone. So you can see this is the backbone. Here are backbones. Nitrogenous base, adenine, guanine, cytosine, thymine, and we can see that this is the we have the Chargaff rules. According to the Chargaff rules, their amount in the given DNA molecule will be about the same. They are saying that guanine must pair with the cytosine and adenine must pair with the thymine. It is a basically basic rule. Complementary rule is this. So we have the diversity among the living organisms. We have a lot of diversity into the human genetics, and you know that our DNA is basically, we can say that we have this structure of the DNA. This is the linear structure. You can see you have a string of the DNA. So they are forming to the human body. They are basically thread-like structure. So what will happen if we just basically, you can say, if we just leave them like a thread into the human body. So we, like our own lifestyle, we arrange things in our surroundings. Same, our body arrange things for ourselves, for our own body. So DNA is basically wrapped around the histone proteins. So what are the histone proteins? You can see the DNA is basically wrapped. These balls are basically histone proteins. When your body needs replication, these are basically unwrapped from the histone proteins and replicate, and again is basically wrapping. These are the highly alkaline proteins found into the eukaryotic cell nuclei that package and order the DNA into the structure unit called the nucleosome. Histone are a group of the basic protein that is associated with the DNA and have the DNA to condense into its chromatin. So we have the genetic information about the DNA, which is basically DNA functions. We know what about the genetic blueprinting. So blueprinting means that DNA genetic material which carries all the hereditary information. We all know that our all hereditary information, it's mean what's coming from my father's side and what's coming from my mother's side. That is basically my inheritance. So you can say that is basically my hereditary. And that is basically transferring from, some of the traits are transferred from my mother's side and some are from my father's side. So same in the case when I will have kids, then I will transfer some of my traits into them, and that would be from my mother's side and from my father's side. So this is basically DNA, or you can say genetic blueprinting. So we have replication. Replication meaning DNA have to replicate, or you can say it has to increase into the number. Into the number. This is essential for the transfer of the genetic information from one cell to its daughter cell. As I told you that when replication take place, one trait from the mother side, one from the father side, and when they combine, they will give rise to another structure that is basically the replicated structure, and that is a newly formed structure of the DNA. We have the chromosome. DNA occurs inside the chromosome. This is essential for the equitable means equal distribution of the DNA during the cell division. Then we have the recombination. Recombination means cross-oving the new combination. It's mean we just during meiosis, crossing over give rise to new combination of the genes called the recombination. If we focus on the meiosis, during the meiosis threads, they basically exchange the material. Same in the case when we talk about some genes from the mother side, some genes from the father side. That is the recombination, or the recombination, the joining of the two different traits or two different genes, and that is basically recombination, giving rise to the new combination. Mutations are the mutation basically causes the evolutions. You can say any change into the nitrogenous base sequence can cause the mutation. I have already told you, but what is the mutation and how it can cause the variations or the evolution. So first then we have 6 number. We have the transcription. DNA gives rise to the RNA through the process of the transcription, catalytic activity of the DNA. What do they want to say? They want to say that during transcription, your DNA give information to RNA. That is basically code from the DNA for the formation of the proteins. Cellular metabolism, it controls the metabolic reaction of the cell through the help of specific RNA synthesis of the specific proteins, enzymes, and hormones. We have differentiation due to the differential function of some specific region of the DNA or genes. Different parts of the organism get differentiated in the shape, size, and function. As we all know that we have different fingers, we have different lines in our hands. Even our fingerprints are basically different. Our hand size is different from the one another. Our eye size, our basically face structure, our length, height, everything is different from the other one. And every part of the body is basically unique in its formation. You can see some can have a joint fingers. That is again differentiation from the normal finger formation. So it is again the differentiation. So we have the development. DNA controls development of the human body through the working of an internal genetic clock with or without help of the extrinsic information. What is the extrinsic information? It's mean the simplest one is this. When the baby is born, we all know that it has to grow. It has to be grown into the adult. So we don't give him, or we just don't stretch their baby, or we just don't give him any factor, or we just don't give him so much things कि भाई ये बड़ा हो जाए, बेबी जल्दी से बड़ा हो जाए. टाइम टेन्योर है, ठीक है, 1 माह 2 माह 3 माह. अब हम उसको 1 माह में मतलब कि हम ये नहीं चाहते कि वो बिल्कुल ही यानी कि 18 साल का हो जाए या 20 साल का हो जाए. So DNA genetic clock, it's mean your genes are basically storing all the information from your born to your death. Even DNA also store that कि किस टाइम पे आपको कोई डिसऑर्डर भी होना है. सो ऑल इंफॉर्मेशन आर बेसिकली स्टोर्ड इन द डीएनए जेनेटिक क्लॉक में. अगेन कि आपकी डेवलपमेंटल स्टेजेस हैं जो कि डिपेंड करती हैं कि 1 माह में आपके हाथ कितने बड़े होने हैं, आपके पाँव कितने बड़े होने, बॉडी का साइज कितना होना है, ठीक है. एंड आल्सो डिपेंड्स ऑन द फैमिली. अगर आपकी फैमिली में सब लंबे कद के हैं तो डेफिनेटली उस हिसाब से आपका कद बढ़ेगा. छोटे कद के हैं तो उस हिसाब से बढ़ेगा. एंड इन द वेरिएशन केस द डीएनए म्यूटेशन वो एक अलग से केस हो जाएगा कि सब छोटे हैं लेकिन आपका कद लंबा है. दैट इज अगेन डीएनए म्यूटेशन. So DNA fingerprinting. Hypervariable microsatellite DNA sequences of each individuals are distinct. They are used to the identification of the individuals and the deciphering their relationships. The mechanism is called the DNA fingerprinting. You can see we have the different DNA fingerprints into our in our fingers. Every person has the different lines in their fingers, thumb, everywhere. So we all have the different retina too. Gene therapy. Defective hereditary can be rectified by the rectified mean. If you have some defects that is basically transformed from your parents, that can be rectified, that can be correct by incorporating the correct genes into the place of the defective one. That is basically called the gene therapy. Gene therapy पे आजकल बहुत ज़्यादा काम हो रहा है क्योंकि जैसे कि हमारे पास डिजीज है सिकल सेल एनीमिया की, ठीक है. सिकल सेल एनीमिया एक ऐसा डिसऑर्डर है जहाँ पे सिंगल अमीनो एसिड सीक्वेंस की वजह से पूरा आपका जो ब्लड का सीक्वेंस है, ब्लड सेल फॉर्मेशन है, वो सारी आपकी डिफेक्टेड हो जाती है. जिसकी बेस पे हमने साइंटिस्ट ने उसके ऊपर जीन थेरेपी, यू कैन से कि उसको एक आर्टिफिशियल अमीनो एसिड का सीक्वेंस बनाया, उसके पुराने सीक्वेंस को देख के और उसको इंसर्ट करने की कोशिश की, जिसमें वो किसी हद तक कामयाब भी हुए हैं. दैट इज द बेसिकली जीन थेरेपी कि उसको बेसिकली एक अपनी मर्जी का वी डोंट से अपनी मर्जी, वी कैन से कि हमने उसको जो पहले सीक्वेंस था जीन का जहाँ पे वो डिफेक्टेड था वैलिन, ठीक है, तो वैलिन को हमने देखा, उसको स्ट्रक्चर हमने देखा, उसकी अरेंजमेंट हमने वहां पे देखी जो कि ऑलरेडी पहले ह्यूमन सेल्स के अंदर थी, आरबीसी के अंदर थी. उसी को पढ़ के हमने आर्टिफिशियली डिजाइन किया अगेन नया वैलिन और उसको इंसर्ट करवाने की कोशिश की उसी जगह पे जहाँ पे आइसोल्यूसिन आके लगा था उसकी जगह पे, ठीक है. तो ये एक एग्जांपल है जीन थेरेपी की. तो डिफरेंट डिजीजेस में जीन थेरेपी यूज हो रही है. उससे पहले हमें इनकी डिटेक्शन करनी पड़ती है. डिटेक्शन के लिए हम डिफरेंट लैब एक्सपेरिमेंट्स करते हैं. दैट इज एन अदर वर्ल्ड, सो आई एम नॉट गोना डिस्कस इट हियर. वी हैव द एंटीसेंस थेरेपी. एंटीसेंस थेरेपी इट्स मीन द एंटी-mRNA और द एंटीसेंस RNA will therapy. We can say it can have you can say to the interference RNA therapy. We use into the pathogenic infections that are basically from some microbes. We all know that why we use the antisense therapy. We all know bacteria have their own RNA, virus have their own RNA. So anti-mRNA it's mean कि जब उनकी प्रोटीन्स बनेंगी तो उनकी डेवलपमेंट होगी, ठीक है. तो जब हम एंटीसेंस थेरेपी यूज करेंगे जहां पे हम उसकी आरएनए की प्रोडक्शन को बेसिकली mRNA की mRNA को फर्दर प्रोटीन के बनाने से स्टॉप करवाएंगे, तो ऑब्वियसली जब उसका प्रोटीन नहीं बनेगा तो उसका स्ट्रक्चर नहीं बनेगा, जब उसका स्ट्रक्चर नहीं बनेगा तो डेफिनेटली उसकी बॉडी डेवलप नहीं होगी। तो यह इसलिए हमने इसको पैथोजेनिक जीन्स को उनका एक्सप्रेशन बेसिकली लो डाउन करने के लिए हमने उनको यूज किया है। जैसे कि कोई भी बैक्टीरिया इन्फेक्शन है तो उसमें डेफिनेटली बैक्टीरिया के अंदर भी जीन्स है, ठीक है, तो उस चीज का एक्सप्रेशन हमने बेसिकली सप्रेस करवाया है ताकि फर्दर डिजीज को एनहांस ना करवाया जा सके। So we have the structure, RNA structure, we have the DNA structure, the common one. So I'm not going to talk about all much on this. So we can have three structures of the RNA. You can see the sequence of nucleotide, primary, secondary, the hairpin loop. You can say hairpin loop is here. You can see the loop is basically anticode, and you can see loops are basically forming. And the next one is basically 3D structure. So we have the types of RNA. We have the messenger RNA carry genetic information copied from the DNA in the form of the 3 base code, each of which specify a particular amino acid. Then we have the transfer RNA के RNA से messenger RNA से information take करता है, transfer RNA जो कि उसके पास transfer होती है, उसके बाद एक intermediate RNA है जो कि information को read करता है, और transfer RNA further इस information को ribosomal RNA के पास भेजता है ताकि वो polypeptide ताकि वो प्रोटीन की फॉर्मेशन करवा सके, कि प्रोटीन के जो स्ट्रक्चर्स हैं वो बन के ह्यूमन बॉडी से बाहर निकल सके, ठीक है, सॉरी सेल से बाहर निकल सके। सो सॉरी, वी हैव द आरएनए फंक्शन. So function, it is the DNA by serving as a messenger to relay the proper genetic information to countless number of the ribosome in your body. The other main function of the RNA is to select the correct amino acid needed by the each ribosome to build new proteins. As we have known that messenger RNA it कोड्स इन्फॉर्मेशन, डीएनए से प्रॉपर इन्फॉर्मेशन लेगा, ट्रांसफर आरएनए को ट्रांसफर करेगा और राइबोसोमल आरएनए फर्दर उसको रीड करके एक प्रोटीन बनाएगा। तो इन सबका फंक्शन ये है कि दे हैव टू वर्क करेक्टली। एक भी गलती हुई तो गलत प्रोटीन या मिसफोल्ड प्रोटीन और यू कैन से डिफेक्टिव प्रोटीन का स्ट्रक्चर बिल्डअप हो सकता है, जिसकी वजह से आपकी स्ट्रक्चर अरेंजमेंट बॉडी की डिफरेंट हो सकती है। So we have genetics. In the genetics, we have the genes, we have the traits, a segment of DNA that codes for a specific trait, and the trait characteristics of an organism can pass on to its offspring to the DNA. So what is the gene? That is basically code for a specific trait. So we have, you can see we have gene, and above gene we have trait. Gene concept, hereditary describe how some traits are passed from parents to the children. The traits are expressed by the genes, which are small sections of the DNA that are codes for the specific traits. Genes are found onto the chromosomes. Humans have two sets of the 23 chromosomes, one set from the each parent. We have different category of the genetic information, that is basically allele, gene dominant, recessiveness, homozygous, heterozygous, genotype, phenotype, and the Mendelian inheritance. We can say what is the allele. We can see these all are basically alleles: A, B, C, D, E, A, B, C, D, E. Different in every organism. There are different possibilities of the given traits. Every trait has at least two alleles, one from mother and one from the father side. We can say eye color, brown, blue, green, hazel. Dominancy. Dominancy, you can say here is the dominant trait that is the mask, you can say the appearance of the recessive gene. First कर ले कि एक पेरेंट्स में से एक की जो आई का कलर है वो ब्लू है, एक की आई का कलर है वो ब्राउन है, लेकिन क्योंकि फैमिली में ज्यादातर लोगों का कलर ब्राउन है तो डोमिनेंसी क्या होगी कि ब्राउन कलर का ही आगे से ऑफस्प्रिंग आएगा आई का कलर, ठीक है। ये है डोमिनेंसी कि वो ब्राउन कलर बेसिकली डोमिनेट हो रहा है बाकी ब्लू कलर पे, ग्रीन कलर पे, जो भी कलर्स हैं, ठीक है। तो दैट्स वाई वो फर्दर ब्राउन कलर को ही मोस्ट प्रोमिनेंट करवाएगा, ठीक है। जैसे कि हमारे बहुत सारे एशियंस हैं, वी ऑल हैव द ब्राउन एंड द ब्लैक कलर्स ऑफ आवर आईज। हमारे में से बहुत कम लोगों के जो कलर्स हैं वो डिफरेंट हैं एक दूसरे से आई कलर के। सो रिसेसिव में क्या होता है कि रिसेसिव में अगर दोनों अगर एक साथ यानी कि होमोजाइगस के तौर पे सामने आए और फर्दर जो है dominant gene को basically mask कर दे, ठीक है। The recessive trait may be expressed when the recessive genes are in the homozygous condition or when the dominant gene is not present, ठीक है। So अगर फर्स्ट कर लें कि फर्स्ट में हमने पढ़ा था कि B और B was present there, so the structure of the color was the brown, so here are the blue, so it is the basically, so यहाँ पे blue color dominant होगा क्योंकि हमारे पास यहाँ पे dominant trait यहाँ पे present नहीं है, that's why उसका color पे further blue हो जाएगा आएगा, ठीक है। Heterozygous, a diploid organism is heterozygous at a gene locus when its cells contain two different alleles of a gene. You can see we have the B, small b and large b, small b and large b. We can have father genes, mother genes, we can have father genes. Homozygous alternative form of the given genes are called the alleles, and they can be dominant or the recessive. When an individual has having the same alleles, whether dominant or the recessive, they are the homozygous. You can see RR. See, it can be the round one, wrinkled one. Genotype which means that is basically genetic appearance, the entire genetic identity of an individual that does not show any outward characteristics, for example gene and mutation. इसका मतलब है कि ऐसी कोई characteristics जो कि आपकी बॉडी के अंदर हुई है लेकिन वो apparently आपको शो नहीं हो रही है, ठीक है। जैसे कि मैंने आपको सिकल सेल एनीमिया की एग्जांपल दी थी, इस केस में भी आप देख लें कि apparently जीन का सीक्वेंस नॉर्मल चल रहा है, ठीक है। ये देखिए, ये आपको एक नॉर्मल सीक्वेंस चला रहे हैं। अब आपको बता रहे हैं कि यहाँ पे एक हमारे पास म्यूटेशन है, ठीक है। ये जीन ए है और ये जीन बी है। वी कैन सी वी हैव अ सीक्वेंस दैट इज बेसिकली रनिंग हियर, बट हमारे पास यहाँ पे बेसिकली वो आपको बता रहे हैं कि यहाँ पे म्यूटेशन कॉज होगी बिकॉज दे हैव हाईलाइटेड दैट एरिया कि ये वो एरिया है जहाँ पे म्यूटेशन कॉज होगी, जिसकी बेस पे फर्दर उसकी जो जीनोटाइपिक है, जो जीन्स का सीक्वेंस है, वो चेंज हो सकता है। Also we have phenotypic, you can see brown red versus the white and the brown one, कि brown red था और मतलब कि brown father and white mother, so their combination can give rise to the white and the brown red. So we have, and with our slide we have some reference, you can read further from the books, the Satyan and then we have the Langer, we have other books of the biochemistry. So thank you so much for listening and thank you so much for your cooperation.